How organisms and their cells can switch on and turn off genes is complicated. In order to understand complicated phenomena, we generally look at the simpler examples, study the phenomena, and then the lessons we learn, we can apply them to more complex situations. So this is no different. First of all, let's see how a simple organism, a bacterial cell, can turn its genes off and on or off. So let's look at this example. E. coli transcription regulation was worked out by Francois Jacob and Jacques Monod. They basically were studying E. coli and they came across a system of genes which is responsible for processing lactose. Let's see the anatomy of these genes, how they are arranged on the bacterial chromosome. So we have structural genes. Structural genes meaning when the messenger RNA is made from them, they are converted into a protein. We have a regulatory element adjacent to it. Upstream, as I mentioned earlier, upstream means towards the 5' end of a particular segment. So upstream of this would be in this direction. So upstream from the structural genes is special sequences on DNA. These are not converted into messenger RNA. They have a regulatory role. These regulatory sequences basically influence the structural genes when these genes will be turned on. So the regulatory sequence has two elements, a promoter. This is promoter, as we have seen earlier. Promoter is the region of DNA. It is a sequence of nucleotides to which RNA polymerase binds. Here we have an additional sequence called operator. Operator is the region between promoter and the structural genes, which basically has a sequence to which a regulatory protein or a regulatory entity can bind. Upstream from this region is another segment of DNA which is making genes which has genes that encode for proteins that can influence, that can bind the operator. So this is regulatory gene and this is the promoter of this regulatory gene. Lac operon basically, or an operon is a sequence, regulatory sequence of a particular gene, and operon basically is the regulatory sequences plus the structural genes. So this is our lac operon. So this is the anatomy of the operon. We are going to study how this functions. Reason it is important is that for bacteria, they prefer glucose as a source of energy, as a molecule they can degrade and extract energy from. E. coli lives in our digestive tract and we are not always eating glucose. Sometimes we eat lactose, which is the sugar which is present in milk. Lactose is basically a disaccharide made up of two monosaccharide units, galactose beta-linked with glucose. So this sugar, if lactose is present, bacteria would need to make enzymes that can process the lactose sugar. If lactose is not there, it will be very wasteful for bacteria to keep on making enzymes for a substrate which is not present or available to this organism. So let's see how this operon works. If there is no restriction, RNA polymerase can bind the promoter and it can transcribe the genes, the structural genes we have talked about, without any problem. However, this happens when the lactose is present in the environment of this bacteria. When lactose is not present, there is a protein a repressor protein that can bind the operator at this region, and it prevents RNA polymerase from interacting with the promoter and from doing the from transcribing the structural genes. So this is the situation when lactose is absent. When lactose is introduced into the system, what happens is lactose binds this repressor. When lactose binds this repressor, we have already seen allosteric or how conformational changes occur in enzymes, when we were talking about enzyme regulation. So here the principle is the same. When lactose is present, it changes the conformation of this repressor. When that happens, repressor can no longer bind the operator. Repressor has two domains, special domains. One can bind the DNA at the operator region. Other domain binds the lactose molecule. So here, when lactose is present, the repressor comes off the DNA and RNA polymerase can now freely bind the promoter region and transcribe the structural genes. Here, since lactose is inducing the production of these genes, which are used for lactose metabolism, it is referred to as the inducer. And the inducer basically, when binds the repressor, it releases the repressor from the DNA and transcription can take place. So what are those genes? The structural genes, one of them is beta-galactosidase. It basically is the enzyme that breaks the disaccharide into two monosaccharide subunits. Permease is the protein that allows or facilitates the import of lactose in the bacterial cell. Transacetylase is also a protein that helps in processing this lactose. So we have seen what happens when lactose is present in the system. So we have just seen what happens when lactose is present. Bacteria, our E. coli, can induce the production of enzymes and other proteins that will assist it in processing lactose. So we are going to see another example of transcription regulation in this system, in bacterial system, E. coli system, in the next module.